Hi, everybody.
Hello.
Can you guys hear me okay?
Yes.
Hi, can you guys hear me?
Yeah,
Yes,
hear
we
you,
can.
Rachel. Yes.
Okay, awesome. I had at least my speakers on something different. Okay. All right. So I think we have everybody that we need here. So I think we can go ahead and get started. I'm going to share my screen first. All right, can you guys see that slide okay?
Yes.
Okay.
Yes.
All right. So, hi, my name is Rachel Wilbur. Thank you for coming to the Graduate Research Colloquium. I'm going to be your moderator today for this session 7. Just a couple of ground rules before we get started. Each contestant will have about 12 minutes to present. Your time will start the second you start speaking. I will give you 1 minute warning by interrupting your talk over Zoom. Once you are ready to present, give me a viewable cue like start or ready or something like that, and I will advance through your slide deck. During the presentation, give me a verbal cue like next, advance, or proceed to advance to the next slide. I will be introducing you before you talk. And that way you can just jump directly into your presentation and you won't have to worry about the introduction taking up any of your 12 minutes. So any questions before I introduce the first contestant and they start going through their presentation? Okay, so I'm going to go to the next slide and then just going to briefly introduce them and then they will get started. So we are going to start with, and I apologize if I pronounce your name wrong, Sagar
It's
Umesh.
okay.
Okay. So I'm going to let you go ahead and get started, okay? As soon as you start talking, I'm going to start my timer and just let me know when you want me to move the slides.
Okay, I'm ready. You can
Okay, go ahead.
Thank you for the introduction. Next. Hello, everyone. So this is the outline of my presentation. So I'll start with the motivation, the background of the project. Then I'll move on to the objectives, the molecular modeling work, some of the findings I have so far, and I'll end the presentation with acknowledgement. Next. Yeah, so the slide shows the image of the Mars surface taken by one of the NASA rovers, which is a machine. And so in recent years, manned mission to Mars and deep space exploration has grabbed a lot of attention from public researchers from universities, industries, space agencies like NASA, SpaceX, which is quite evident from all the news in the news outlets. Then there are so many fictional movies, TV series exploring this deep space exploration concept. And then there's, the time to time Twitter feeds by Elon Musk himself. But manned mission to Mars is not a reality yet. For one, we need space vehicles that can carry humans along with, all the necessary instrumentation to the red planet, as well as that can sustain extreme environmental conditions. Next. Therefore, the motivation for this research is simply the need for lightweight, ultra-strong materials for manned mission to Mars, because the current state of the art material is unable to provide this requirement. Next. So carbon nanotubes have been around since decades now, and they are the key to achieve this. CNTs have much higher strength and stiffness than traditional carbon fibers. But for the past 20 years, the effective use of carbon nanotubes in composite has been slowed, simply because of poor polymer to CNT load transfer, excessive CNT agglomeration, as well as lack of higher production rates for CNTs. Next. Then the recent fabrication of flattened carbon nanotubes at Florida State Universities gave us a whole new perspective towards looking at the traditional CNTs. So they observed that upon stretching these CNT films, they are able to self-assemble themselves into flattened arrays so that we can pack tightly and more reinforcement into the composite. Next. Next. So, but, and therefore, because we can pack more and more reinforcement, maybe this composites might be, more optimal than the traditional carbon nanotubes composite that we are using right now. Also, they can offer higher chemical reactivities. But what polymer to choose even to make this polymer composite is yet to be explored. Maybe, there's a unique combination of polymer and flattened CNTs that can offer high load transfer, but also optimal mechanical properties. This may also reduce, the production cost, and we can manufacture composites, in a panel size sheet size samples. All of these questions are yet to be answered, and therefore computational modeling can provide quick and inexpensive answers to all of these questions and much more. Next. That's why, and even the huge composite literature shows that in order to improve the load transfer, it is necessary to target the interface between this polymer and CNTs. And therefore, the objective of this research is simply to design molecular dynamics simulations to address that interface. So towards that, we've designed 3 different simulations that test this interface in 3 different scenarios. So first is simply to see the interaction of different polymers with the flattened CNTs. The second is, how the interface behaves when it is sheared or pulled in the axial direction, and as well as separation resistance as to how this interface behaves when it is pulled in transverse direction with relative to the polymer surface. Also, these simulations can be used as screening tools to compare different polymer systems interacting with these flattened CNTs. So in today's presentation, I'll be presenting on 5 different polymer systems, 2 cyanide esters, which is a thermoset, 1 part thermoset fluorinated and non-fluorinated. Then we have polyimides, again, fluorinated and non-fluorinated, and then PEEK. Next. Next. So these are the MD simulation setup. So first we have the interaction energy, at which the purple layers are the flattened CNT layers, and then we have polymer in between. Next. Then in the second simulation, which is a shear resistance, in which we pull 1 flattened CNT layers in the axial direction as shown by the arrows relative to the polymer surface. Next. And then finally, we have the separation resistance, where we pull the flattened CNTs or the entire simulation box in the transverse direction as denoted by the arrows. Next. So these are the simulation details. The molecular dynamics simulations are set up and ran through LAMS MD software. One of the key features of any molecular dynamics simulation is the choice and implementation of PCFF of the force field. A force field describes the interactions between atoms within the within the molecules itself. So it is very necessary, and it influences the results a lot. In this case, I'm using polymer consistent force field, interface force field, which is proved to accurately describe these interactions. The model size varies from 30,000 to 60,000 atoms, which is quite a lot, quite big models. The polymerization is simulated using fixed bond react command, which again significantly reduces the computation time of these simulations. And then we've created different models with varying mass fraction as shown on the slide. Next. Now jumping onto the results. So first we have the interaction energy on the y-axis as a function of varying mass fraction on the x-axis. So on the y-axis, the higher negative value suggests better interactions. So as you can see on the chart, for all of the 5 polymer systems, the interaction energy shows a similar trend, which offers us a qualitative validation of a kind. Then for lower mass fraction, the interaction energy is lower simply because there's just not enough polymer to separate the flattened CNTs completely so that we can take advantage of that surface area. Then as you increase the mass fraction, the interaction energy increases. And then once you reach a sweet spot, around 40 to 45%, as can be seen from the chart, the interaction energy remains constant, which is again similar for all of the 5 systems. Next. So the main takeaway from this graph is that, PEEK and non-fluorinated polyimides offer better interaction. That is simply because they have this ability to align flat with the nanotube surface, and therefore there's a better interaction within the stiff backbone, aromatic backbone that they have. Next. Then we have shear resistance, where we have the log of friction force on the y-axis as a function of velocity on the x-axis. Now the polymer results are plotted against two cases of pure flattened CNT results. So the first, right at the top, is the commensurate flattened CNTs, which is a perfect alignment between two layers of flattened CNTs. As you can see, it has the highest friction at lower velocities, realistic velocities. But the commensurate flattened CNT case, where you have this perfect alignment, is practically unachievable. You cannot really achieve that perfect alignment. That's why we have composites. Then the second case is incommensurate CNTs, right at the bottom, where you have a slightly disalignment. And as you can see, it dramatically reduces the friction force. But as soon as you add the polymer between those incommensurate CNTs, it significantly increases the friction force. And then contrary to the interaction energy, in this case, right at the top, fluorinated polyimides offer the highest friction of all the polymer layers. That is simply because of the presence of the fluorine groups offering steric hindrance. Next. So the main takeaway is commensurate flattened CNTs, which is unable to achieve, offers better friction, and then among the polymers, the fluorinated polyimides is the best to use. Next. Then we have separation resistance, as can be seen on the graph, where you pull the simulation box in transverse direction. Now, in this case, almost all the systems shows again similar trend. Next. So the main takeaway is, so the highest peak strength is shown by cyanide esters. Peak, they again offer highest stiffness, simply because they are really small molecules. So you have better infiltration among the CNTs. And therefore, it is really hard when you separate them apart. And if you look at the stiffness, it's nearly the same for all of the systems. Next. So in conclusion, next. Next. So we have 5 different comparisons between 5 different polymer systems. Next. Next. Peak, polyimide, and cyanide esters. Next. Of which Peak offers the highest and strongest interaction of them all because of its ability to align flat with the flattened CNTs. Next. Then we have polyimides, which offer strongest shear resistance simply because they are very large molecules and offer steric hindrance because of their respective chemical groups. Next. And then we have cyanide esters that offer highest separation resistance. Next. So to conclude, in my subjective opinion, non-fluorinated cyanide esters may be the best choice of all the polymer systems to use for this application, simply because, as I mentioned, they are relatively easy to process because it's just a one-part system. It's a small monomer, so we get better infiltration between the nanotubes and this polymer, as well as they offer excellent separation resistance and moderate interaction energy and shear resistance. But now this is just my opinion. And that's why all of these plots that I've shown can be used as design charts by experimentalists and people in the industries who actually manufacture these flattened CNTs to see and decide what polymer system to choose from. Currently, we are populating this graph with lots more data so that it's easy for people to look at and make their own decisions. Next. So in the end, I would like to thank judges for their valuable time. Of course, the GSG team for arranging this event. This research is funded by NASA Space Technology Research Institute for Ultra-Strong Composites by Computational Design, led by my advisor, Dr. Greg Odegaard. It is a unique collaboration between experimentalists and modelers from 11 universities to aerospace industries. All of the simulations are performed using superior high-performance computing cluster here at Michigan Tech. And it's a massive task. It's not a one job of one person or one team. We need to come together to achieve this. And I hope my research sends a message that, in order to advance technology to a point where people can practically use it, scientific collaboration is absolutely necessary. Thank you. Next.
All right. That brings us to the end of your time. And I want to apologize. When you were on your conclusion slide, you had a minute left, but I felt like it was wrapping up, so I didn't want to interrupt you there.
Oh, it's okay.
Okay, so next we have 5 minutes, I believe here, yes, for the judges to ask any questions that they have. Let's see. So it says here in the guidelines that each judge should have at least time to ask at least one question. So I don't know if the judges just want to kind of jump in here, if anyone wants to volunteer to go first with their questions, but now is the time to do that.
Can I have a quick question about the result you show?
Yeah.
The picture you show for the comparison is simulation results or experimental results?
These are all simulation results.
Yeah, that's what I thought, because you use superior, right? High performance computing. What the numerical method you use for the simulation? Do you know that?
Yes. So I've directly used molecular dynamics, software, molecular dynamics, simulations, and therefore the force field, which is, PCFF-IFF, the polymer consistent force field, inbuilt, it inbuilt has all of the interactions and that numerical coding to it.
So do you have some idea what actually the numerical method coded in this software? It's finite element, finite difference, or integral equation method?
Yes. So it's definitely, because it's a nanoscale, so it's definitely not, any of the finite elements or finite difference methods, because, difference in the length scales, right? They are mostly used for microscale. So in this case, yes, for calculations, it uses algorithm called as Worley's algorithm, which is an iterative algorithm. So it takes into account the initial condition of the model that you provide, the temperature and the size and the velocity of the atoms. And then it iteratively calculates all of the properties that you want it to. So it's automatically, the Worley's algorithm is automatically coded into LAMP's software package.
Another quick thing is contribution is mainly the comparison of different methods, right? Is there any other study in literature also did a similar thing?
So I'm just comparing different polymers. The simulation details are same for each system to compare them. And literature, the simulations that we designed are informed by the literature as well. But we did not have any direct comparison to anything in the literature.
Okay, thank you.
Thank you.
Hi, Sagar.
Yes.
Thank you for an excellent presentation. I love learning about composite materials and new applications.
Thank you.
I just was wondering, based on your results, then where would you propose this project go next?
Yeah, so that's an excellent question. So the work that I've presented today is published. And currently, as I mentioned, that these properties these are at nanoscale, right? So you cannot really use this results. But so our collaborators, which work at, much higher length scales, like, micro scale at University of Colorado and University of Minnesota, they are actually using all of these results into their model, which is, at mesoscale, to scale up these properties and, determine the effective properties of the composites. That is used, the experimentalists at, MIT and Florida State to decide what polymer system to choose. So to giveaways, today I'm presenting this. So definitely these are the systems that are not used for the actual application. Because that's a proprietary information. But this, the data that I've shown so far has definitely made, helped them to make that decision. So that's how it's scaled up the scale.
Great, thank you.
Thank you.
Hi, can I ask a question?
Oh, yes, please.
Hi, Sagar. Congrats, it was really good.
Thank you.
I was wondering in slide 10, you showed the interaction energy, and I was wondering what that was negative, right? What does that mean, a negative interaction energy?
Oh, so that's simply because, the way we calculate, it's a van der Waals forces, basically. So it just shows the negative, it just how it is calculated using LAMP's software package. So, and, the negative sign just implies that there's an attractive force.
Okay, I see.
Yeah,
Okay,
that's just how it's calculated.
And in slide 11, there were like velocity values on the x-axis, right? And I think they were pretty high, from 300 to 500
Yes,
per second.
yes.
What does
So
that mean? I mean, how do you induce velocity?
Yeah, so can you go back to that slide, please? Yeah, that's true. Like, realistically, we cannot really have that high velocity. Experimentalist doesn't use that, but that's the beauty of computational modeling, right? We can simulate extreme cases in this case. So we just chose to use those velocities so that, we can get the trends of friction force versus velocities, which is a logarithmic dependence, right? So as professor asked me about comparison, with literature. So that's the qualitative comparison that we could do. We found data in the literature where we had friction force versus velocity plots. So to compare our results, we just simulated those velocities just to qualitatively compare them. But yes, realistically, we cannot really use those.
Okay, thank you.
That's just for comparison.
Okay, so we have some time now for the judges to do their scoring, unless there are any other questions from the judges. So if there are no other questions from the judges, we can continue to take public questions until the judges are ready. So judges, just let us know when you're done. Otherwise, any more public questions are more than welcome.
Hi, Rachel. I'm done with evaluation. Thank you.
Okay, thank you so much.
Yeah, Rachel, I'd also sent you a private message, but I'm also ready.
Okay, yeah, for some reason when I'm screen sharing, oh, there, I see now where the chat comes up. All right, let me just make sure there's nothing else in the chat here that I need to go over, and then we can move on. Let's see. Okay. All right. So we are going to then move on to our next presentation. If I get the chat window out. Great, my Zoom is freezing. If you guys can hear me, I'm sorry, but Zoom is freezing here.
I can hear you.
You
but
can? Okay, my screen is still not working, so I'm hoping in a second here it will.
So the screen doesn't work well. There is a black square on the slide.
Rachel, do you want to share? Should I share it from my end?
Yeah, maybe, because it says, even though my microphone's working, it says Zoom is not responding and it's going to make me shut down here.
All right, give me a minute. Sorry for the logistical delay, everyone.
Yeah, sorry about that, guys.
Can you stop your share so that I can start sharing?
I think I'm going to have to completely shut down my computer in order to do that. So I'll come back in the next 5 minutes and take over from you.
Oh, all right, that sounds good.
All right, I'll see you in a bit.
Okay. All right, can you guys see my screen now?
I can.
Not yet.
Not yet, no.
Still no?
Yeah, I still see Rachel's frozen screen.
All right, I'm gonna do one thing.
Okay, now I can see it, Shibata.
All right, so let's just directly dive into your presentation.
Yeah, that sounds good.
Good morning, everyone. I would like to introduce our next speaker, Jessica Bruning from the KIP department at Tech. Her topic of presentation is microbial-derived short-chain fatty acids and salt-sensitive hypertension. Whenever you're ready, you can begin, Jessica.
All right, thank you, Shibata. Good morning and welcome to my talk. I'm going to be discussing the microbiome, its metabolites, and how they're related to salt-sensitive hypertension. Next. To discuss this multifaceted, exciting story, I'm first going to have to introduce hypertension and the microbiome, followed by the rest of the whole outline, hypothesis, aims, conclusion. Next. Hypertension is defined as a chronic condition of elevated blood pressure 130/80 millimeters of mercury. And even though this pathology contributes to approximately 9.4 million deaths a year globally, very little is known about underlying mechanisms that lead to hypertension. What is suspected is that hypertension is a multifaceted pathology influenced by lifestyle choices such as diet, the microbiome, its metabolites, and genetics. Next. And can you give me 2 clicks, Shivata, please? Thank you. So long-term blood pressure regulation, 1 more click, is influenced by 3 different systems: the autonomic nervous system, the kidney, and as well as hormones. And what all 3 of these regulation systems have in common is the brain. Of particular interest to me and my team is the paraventricular nucleus of the hypothalamus, abbreviated as the PVN. And specifically in autonomic nervous system regulation and as well as the kidney, we have an increase in what's called sympathetic outflow from the brain to the rest of the system. And these events cause an increase in mean arterial pressure or blood pressure. Next. What is the microbiome and what does this have to do with hypertension or in host health and disease in general? Well, these little bugs are everywhere. They're all over your body. They're on the chair you're sitting on. They're on the computer you may or may not be touching, and they're on the food you eat. Microbes dominate this planet. And most of the time, they live in a symbiotic relationship with us, specifically the gastrointestinal tract microbiome. They help us obtain micro and macronutrients from the food we eat. But if there's some sort of perturbation in their environment or nutrient availability that they have, it can cause this community to shift. And this shift is referred to as dysbiosis. So maybe one species increases in population density while another decreases. And this dysbiosis has been associated with several pathologies, including schizophrenia, obesity, diabetes, as well as ADHD. Next. And I think some key players in this dysbiosis and the relation to pathology is going to be their short-chain fatty acids. So short-chain fatty acids are bioactive molecules produced by our gut microbiome as a result of fermentation of dietary fibers. If these short-chain fatty acids get into circulation, they have a wonderful opportunity to bind to G protein coupled receptors that exist throughout our tissues. And upon activation, these GPCRs can cause a massive cascade of signals throughout the entire body. Next. So what does our guts have to do with our brain? Well, as you can see in A, if we inject a dye into the guts of these rats, so on the left panel A, this is a spontaneous hypertensive rat versus a normal tensive rat on the right side. If we inject a dye into their guts, that dye can travel via neurons up into the paraventricular nucleus, the PVN, of hypertensive rats more freely. So we have an increase in communication between the gut and the brain in hypertensive animals in comparison to normal tensive animals. We also see in B and C a decrease in tight junction proteins in spontaneous hypertensive animals in comparison to their normal tensive counterparts. So hypertension has an increase in intestinal permeability, allowing for toxins, bacteria, even short-chain fatty acids to enter circulation more freely. Next. And if you give me one click, that'd be cool. So these plots are showing like microbial community composition. The green would be a normal tensive animal in A, and the red is a spontaneous hypertensive rat. And so you can see there's a clear difference between normal tensive microbial community composition and hypertensive communities. And this is also seen in humans. So that's in B. Okay, normal tensive individuals are in green, and hypertensive people, their microbial communities are in red. And you can see clear clustering here as well. Next, please. So what does this have to do with salt-sensitive hypertension? Well, in A, this is another way that we can categorize the microbiome. We just do it by percentage of phyla. So we can see that salt-sensitive animals, animals that you feed salt and they develop hypertension, they're in green. They have a completely different microbial profile than normal tensive animals. And as well, when we feed animals salt, seen in A through D on the right panel, we have an increase in short-chain fatty acid concentrations in their fecal samples, showing that there's some sort of relationship between salt and an increase in short-chain fatty acids. Next. And if you give me two clicks, I think that's enough. Okay. Once in circulation, these short-chain fatty acids can bind to G protein coupled receptors. And as you can see highlighted kind of in the center column in the orange or yellow, these GPCRs are very present in sympathetic nervous system. Okay, next. I'm not going to read all these for you, but there's lots of gaps in the literature. Most importantly, underlying mechanisms through which dietary salt induces hypertension is vastly unknown. And then how these GPCRs can influence neuronal membrane potential has yet to be investigated. Next. So my hypothesis is that an increase in dietary salt leads to an increase in short-chain fatty acids. And then this increase in short-chain fatty acids in the PVN can contribute to salt-sensitive hypertension. Next. Here's just a more pretty image of what I just said. So we get high salt diet that increases short-chain fatty acid production, increases short-chain fatty acids in circulation and in cerebral spinal fluid, leading to an increase in sympathetic outflow and then increase in blood pressure. Next. So I plan to prove this hypothesis model through these three aims: to determine the effect of high salt diet on regional microbial shifts and short chain fatty acids in SD, which are normal animals, and then doll rats, which if you give them salt, they get hypertension or they don't. Next. This is my preliminary data. So in the green, most often than not, researchers will use fecal samples to determine microbial communities. And then I'm showing that intestines and colon microbial communities are vastly different than fecal sample microbial communities. If you give me one click. So this shows that feces are not the optimal sampling unit for microbiome profiling, and we really need to shift our research. Next. My lab has also been shown, if you give me one click, that high salt diet can in fact shift microbial communities. This has been supported in other literature as well. So that is seen not only in the fecal samples, which others have shown, but also we see that in the intestine and the colon microbial associated communities. Next. And if you give me one click. So I did this in SD rats. Now I would like to do it in doll rats. So if you give them salt, they may or may not develop hypertension depending on their genetics. Next. And then after I determine the microbial profiling, I would like to detect short chain fatty acid concentration shifts in animals that are normotensive and hypertensive in plasma and cerebral spinal fluid. Next. Moving on to how the short chain fatty acids will then influence GPCR protein expression. I'll be using Western blot and PCR. I'll be just taking PVN tissue from animals as well as brain slices. Next. And I can also do this experiment, not just in whole animals, but also in cell cultures. So I'll put neurons in dishes, I'll bathe them in these short chain fatty acids, and then I'll also add GPCR blockers and then differentiate how these short chain fatty acids can impact the expression of GPCR on these neurons. Next. And that leads us to my final, if you go back, sorry, the final aim is to determine how high salt diet will influence sympathoexcitatory responses of these short chain fatty acids. And then you could go next too. Thanks, Shavata. So what I've already shown is that these short chain fatty acids, acetate and lactate, do in fact, when introduced into the neuron, will produce an increase in sympathoexcitation. So what you're seeing in this top kind of panel, this is a recording of nerve activity from the peripheral system. So what I do is I hook a nerve in the periphery, and then I inject short chain fatty acids directly into the brain. So I'm showing that brain short chain fatty acids are causing an increase in response in the periphery of the animal here. Next. I'd like to do this in animals fed a high salt diet. So does the high salt cause an increase or a decrease of that systemic response when short chain fatty acids are introduced into their brain? And then I want to prove that this sympathoexcitatory response is in fact caused by GPCR binding and activation by adding blockers. Next. And so here's just my hypothesis model. If the other two were unclear, so we have a rat, we're going to give it high salt diet. That's going to change the microbial profile. That's going to, those microbiome changes are going to change how much short chain fatty acids are produced. Microbes can also influence the intestines. So that's going to increase the amount of short chain fatty acids allowed into circulation and then into cerebrospinal fluid across the blood-brain barrier, increasing short chain fatty acid interaction with PVN neurons. Increased activation of these neurons is going to cause an increase in blood pressure. Next. Thank you, Shavata. So in conclusion, one of my biggest findings here is that fecal samples are not the most optimal sampling unit for microbial community composition, and that short chain fatty acids do indeed elicit sympathoexcitatory responses, and I'm proposing that this is through GPCR activation on presympathetic neurons. And the last slide is just my acknowledgements. And that's, I will take questions. With 30 seconds left.
Yeah, that was pretty timed. I would like to invite our judges to unmute themselves and ask questions to Jessica for the next 5 minutes.
Hi, this is Qigong San. I have a quick question about the result of your research. Are you plan to publish your research result findings in some journals?
Yes, so I am very interested in moving forward with publishing that fecal samples are not the most optimal sampling unit. What I'm waiting for with that data is to collect ion chromatography data showing that short chain fatty acids are also shifted in plasma and cerebral spinal fluid when animals are fed high salt diet in comparison to normal salt diet. So this will just be like a complementary finding to the result of my microbial profiling data.
Thank you. I don't have further questions.
All right, thank you for your question.
Hi, Jessica. It's nice to match a face with an email.
Yes, hi, how are you?
I'm great, thank you.
Good.
So I'm just wondering from the perspective of, this is a very detailed study that's, looking at very specific things. How would this impact human health, or how do you see your results impacting that in the future?
Yeah, so GPCRs are actually like a huge pharmacological target for lots of drugs. And then, so that would be really beneficial, I think, for individuals who do have hypertension and they take like 2 or 3 antihypertensive medications and they still have high blood pressure. And we're just like not sure why that's happening and how to mitigate that. And so if we can find a more precise treatment for them, not just pharmacologically, but also, holistically. So if we can change their diet somehow and change their microbiome, either with prebiotics or probiotics or a combination of those two along with pharmacology, I think we can help decrease how many individuals are suffering with treatment-resistant hypertension. And that would be my goal.
Great. And I'd be happy to answer, or I'm happy to open up to other questions from the rest of the audience.
That was a great question. Thank you.
Hi, I have a question.
Yes.
First of all, a great presentation.
Thank you.
So my question is that in one of the plots, you showed an observable, I think, SS and A.
Yes.
And there was a gap in between. So what does SS and A mean, and what does that gap represent?
Yeah, Shivata, can you go back just a few slides? Right there, right? Is this the slide that you had a question about?
Yes, exactly.
Okay, so SSNA stands for splanchnic sympathetic nerve activity. So splanchnic is just the specific nerve that I was measuring sympathetic activity from. And the splanchnic nerve innervates arterioles that kind of surround our gastrointestinal tract. And these are wildly involved in regulating your blood pressure. So these will determine what's referred to as total peripheral resistance. There are other ways that you can quantify sympathetic nerve activity in relation to blood pressure. The renal nerve is a really good one that I also study. And this is mostly involved in the other half of blood pressure regulation, which would be total cardiac output. So how much like water are we taking back in versus excreting from our body? That's what this nerve's mostly involved in. But the one quantified here is total peripheral resistance. So like how open or closed your blood vessels are is going to determine how much blood pressure exists, right? And so the gap on the bottom is, so on the left side of the gap, this is before the drug is introduced. This is what's referred to as a raw trigger.
I think she lost connection, and that's why she got disconnected.
Okay.
But what we'll do is we will end her session in here. If she joins back and if we have time at the end of the session, we can continue. If the judges are done scoring, we can move on to the next presenter and Rachel can take over from me.
All right, perfect. So do the judges need any more time?
I'm good, thank you.
I just need a few seconds.
No problem.
Dr. Sun, are you ready?
Yes, I'm ready. Please
Awesome.
move forward.
Yeah. Rachel?
Okay, so our next presenter is Manpreet Bora, and he will be presenting on linear and nonlinear optical properties of twisted multilayer TMDCs. So Manpreet, whenever you are set, you can go ahead and I will start the timer when you start speaking.
Thank you, I'm ready. Okay. So hello everyone, I am presenting on linear and nonlinear optical properties of twisted multilayer transition metal dichalcogenides, and I'll be referring them as TMDCs. Next. TMDCs are of special interest because of many reasons. After discovery of graphene, a lot of research interest grew in 2D materials. Graphene is semi-metal and it has closed band gap, but in TMDCs, band gap is present in optical range, that means visible and near infrared region. So that gives us an edge on graphene. Secondly, they lack dangling bonds, which means it is possible to restack these TMDC materials in vertical direction without worrying about their lattice mismatch, which makes them more useful in electronics. Thirdly, they have very strong linear and non-linear properties. So which depends on the stacking we have for monolayers or different layers. So I'll be talking about second harmonic generation in next slides. So in my research, I want to explore the dependence of these properties on the relative twist angle between homostructures and heterostructures of these TMDCs. Next. So transition metal dichalcogenides, TMDCs, they have a formula of MX2. On the left-hand side, the top view shows their hexagonal lattice. And on the bottom figure on the side view, we see that a transition metal, for example, molybdenum or tungsten, is sandwiched between two chalcogen atoms. Chalcogen atoms can be sulfur or selenium. So here these chalcogen atoms are bonded with this tungsten, transition metal with covalent bonding. And covalent bonding is one of the strongest bondings. But when we go from a single layer to other multi-layers in a crystal, we have a van der Waals interaction. If you look at this figure here on the right-hand side, so if we are stacking 2 layers or 3 layers of these TMDCs, we have a van der Waals interaction, and which makes it easier to exfoliate from bulk to single layer. And these materials have band gap in visible range from visible and near infrared region, like 1 to 2 electron volts. And as we go from bulk to single layer, their band gap changes from indirect to direct band gap, which has another plus point. So depending upon the stacking of these, we have common polyforms. One of them is semiconducting 2H and another is metallic 1T. Next. So in my research, I'm using density functional theory to understand their structure, electronic properties, and optical properties, and then experimentally confirming them. So to understand their electronic properties, we need to solve Schrödinger wave equation. And up to helium, we can accurately solve this Schrödinger wave equation. Here you can see that H psi equals to E psi. This simple equation tells you about the energy of a total system. So to go to many body problems, we need some approximation. So we make use of Born-Oppenheimer equation, which basically decouples this wave function of nuclei and electrons into separate ones. And we go from wave functions to electron density using some quantum equations. So here we see that there is one kinetic energy term and one potential energy term, which take care of every interaction. So here comes DFT, which makes the use of going from wave function to electron density. And it says that now we can solve many electron problems to many one electron problems. So now our ground state, it becomes unique functional of electron density. A function means a function of function. So we make use of two properties that if we want to have ground state energy, we can minimize the energy of this function using electron density, and we can get our ground state. So to use that, we use this on the right hand side, we use the schemes, self-consistent scheme, and we start with a trial electron density, and we apply our quantum equations. And if our calculations converge and we get desired electron density and it is equal to a trial, so we say we got our ground state, otherwise it continues. Next. So I have used WASP. It's a commercial software to use density functional theory, and it uses periodic plane wave basis set to calculate ground state energy. So I used two stackings for my calculation. One of them is AB stacking and another is AA stacking. So if you look at the bottom figures, that shows us the side view of these two layers. So these two stackings are the most stable stackings that I was interested in. And if you look at here, this gray atom corresponds to transition metal and these yellow atoms corresponds to a sulfur atom. So in AB stacking, we have some we have some displacement between two layers, like there's some relative twist angle of 0 degree. And in AA dash stacking, we have relative twist angle between two layers of 60 degree. Next. So from my calculations, I got result for band structure. So basically here, these three figures corresponds to energy and momentum relation of TMDCs. So the first figure on the left, it corresponds to a monolayer. If you look at here around this 0, this horizontal line tells you a Fermi level, and this blue curves, they are showing you different bands. So when I'm saying a monolayer is a direct band gap material, I'm saying that we are seeing a direct transition at K point. So a band gap is basically an energy gap, and you don't have any electronic states in this gap. So that means we need this energy to provide. So we need this much energy to provide electrons to jump from this bottom of this valence band to conduction band. So as we go from this monolayer to bilayers, we see that this direct band gap stays there, but we have indirect transition dominating also. So we see that in transition metals, it's the d orbitals which contribute mostly to band gaps. Next. So to understand linear and nonlinear optical properties, we have to look at their dielectric function. So a dielectric function is a complex function which is dependent on frequency. It has one real part and imaginary part. And for linear properties like absorbance, it is related to its imaginary part. So in a monolayer, we saw that there is a prominent peak around 305 eV and a shoulder around 2 eV. We know about the transition that corresponds to these peaks. And as we go to bilayer stackings, we see that there is some redshift in a peak in both stackings, like 2.78 eV and 2.8 eV, but there's also this splitting of a single peak. So by looking at the dielectric function, we can know the stacking that we have for bilayers case, which is very important. And that means, like, if we have this dielectric if we have AB stacking, we are going to expect how much increase in absorbance or decrease in absorbance. Next. So depending on stacking, we have absorbance calculation. Here, absorbance means fraction of photon energy absorbed by a system. So we go from monolayer, which is a black curve here, and to bilayers here. So we see that for a particular stacking, there is more increase at different energies. And so far, there's no study has been done for tri-layer cases. So we have calculated also for tri-layer cases. So this shows that if we have TMDCs and they have, and if we go to bilayers, there is going to be some increase in this region from 2.4 to 2.6. And there is going to be an absorption peak around 1.8 in bilayers. Next. So now looking at nonlinear optical properties. So for a material, its optical response can be explained by this first equation where P is polarization. And these chi terms are suspectabilities. So in a non-linear material, we have these higher order terms, which corresponds to non-linear physics. And we are looking at second harmonic generation and third harmonic generation in these materials. So basically in a second harmonic generation, if we have a nonlinear material and two photons with frequency omega pass through it, we get a photon of frequency 2 omega. That means we are getting photons of higher energy. And these TMDCs, they don't have center of symmetry. And also when we stack 2 layers or 3 layers, there is some change in symmetry, and that induces this second harmonic response. Next. So this second harmonic generation is very important tool to probe your structure, your crystal orientation. So in our lab, we have this setup on left-hand side. So here we use a mode lock laser titanium sapphire to see second harmonic response. So it passes through filter and half wave plate and polarizer, which basically centers its wavelength on this dichroic mirror. And from this dichroic mirror, 50% is reflected to sample and 50% is transmitted. So this transmitted light is our second harmonic response. For example, if we are having, let's say 800 nanometer as our incident wavelength, we are going to expect our second harmonic wavelength as 400. So here we have this lamp, white light to see the structure, and here we have this spectrometer to collect this second harmonic response. So from our first results, we have this polar plot of second harmonic response for WS2 monolayer. If you look at this right-hand side plot, here these black and red colors, they denote parallel and perpendicular component of second harmonic response from the sample that we have. So this shows that it has 6-fold symmetry and it can be used to see the crystal axis orientation. Yeah, next. So far we are using DFT to calculate electronic and structural and optical properties, and experimentally we are fabricating these materials and confirming our results.
1 minute remaining.
Okay. So second harmonic generation and third harmonic generation are very important tools in these days to explore this crystal orientation and their effects with the substrates that we're using. Because in the end, we want them to use in electronics. So we want to understand their effect with substrates. Thank you, that's all.
Professor Charlesworth, would you please go first? Because I have quite several questions to ask.
Sure. My question is a little more general, but what part of this project did you find the most challenging and how did you overcome that challenge?
So I'm an experimentalist, so I had to learn DFT, like theoretically understanding this system, but it was exciting because first I can understand what is the physics and then I can use that physics in lab and verify those results.
Great, thank you.
Thank you.
Great work. I think this is a combination of both a deep theoretical approach and also you also did experiments, right?
Yes.
Yeah, so in terms of theoretical part, when you compute the band structures, right? I believe you use some simulation technique to obtain that band diagram, dispersion diagram. What are the software you are using?
So I'm using WASP. It's a commercial software, but the benefit of using this software is that they already calculate your exchange correlation potentials. So it makes our calculation fast and easy.
Okay.
And accurate also.
In your presentation, you gave expression of the electric permittivity, which depends on the frequency, right?
Yes.
Epsilon, I think you got epsilon omega.
Yes.
So to me, this seems a nonlinear material. You got real part, you also got imaginary part. Usually for a material, it means the absorption, right? Yes. But you didn't show what is your epsilon omega 1 or 2, what that exactly equals to.
So I showed my DFT results. Can you go to slide 6 or 5, please? Back. One more.
Previous one.
Previous one.
Previous.
Yes, this one.
Yes.
So here I use this epsilon omega. Here this is real part, and epsilon to omega, it's imaginary part.
Yes. So do you know what is the exact expression for epsilon
omega?
Okay, it's like omega over like omega square minus omega 0 square or something like that. Do you remember?
So we use Kramer-Kronig relation to get this
Okay.
imaginary part. So does that answer your question?
Oh, okay. You might not remember because whenever you have this relation, probably you have some mathematical functions. So what is exact that function epsilon1 and epsilon2? Do you remember?
Okay, so if we have this kind of relation, so this epsilon, so we first calculate imaginary part. Because from imaginary part using Kramer-Kronig relation, we can calculate real part. So for imaginary part, it depends upon the polarizability per unit area and multiplied with the omega that we have.
Okay. Could you go to the slide with the band structure?
Okay, one more previous slide.
Yes.
This one.
So I clearly see there are some band gaps over there, right?
Yes.
So are these pictures the output of the software WAST you are using?
Yes. So if you look at this first plot monolayer, we have this band gap, which I'm saying is direct band gap of 1.79 eV. And I can measure this experimentally using photoluminescence measurement. So we can measure this direct band gap also. So here, these are DFT results, but experimentally also I can measure it.
Okay. So your experimental result is consistent with your simulation?
Yes.
Okay.
So when we are using DFT and when we are using some softwares like WASP or other softwares and using some potentials in them, they try to underestimate a little bit our band gaps. So experimentally, this band gap is around 2 electron volts, but in my calculation, I got it around 1.79. If I wanted it to make it more accurate, it would be more expensive computationally.
Okay.
Yeah.
All right, so we have about 15 seconds left here for the judge questions.
Yeah, actually, I'm done with my questions. Thank you.
Thank you.
Okay, so we can go ahead and start. We can take some time for the judges to do their scoring. Please let us know when you guys are done. Otherwise, we can be open for any general questions from the audience.
Hey Manpreet, hi, I have a question. Thank you for your presentation, by the way. How long does it take to finish one calculation of DFT?
So, okay, so in this, I'm using, so you know that in WASP, we have periodic boundaries. So I do not need to have big system here. So typically my calculations would be like 3 days or 4 days long, because these are I'm using transition metals in my calculation, and these are very heavy atoms.
Okay, so then what's the typical size, model size that you simulate?
So if I say in terms of supercell, it's 3 × 3.
Oh, okay, so like 100 atoms or so?
No, we just need to have 3 × 3 as our supercell
array. Okay.
Yes.
Oh, okay. And so you mentioned about the exchange correlation function, and you said it calculates automatically.
Mm-hmm.
So is there like the basis, any particular function that you're using?
So yeah, we use this plane wave basis set for calculation, and I'm using PB functional for this. So in that function, basically your internal, your core electrons are frozen. Only valence electrons are included in calculations.
Okay, thank you.
Mm-hmm. Thank you.
Okay, I think we're going to have to go ahead and move on to the next session since we ended up going 4 minutes over. Do we have our next moderator here?
We will end this up Zoom meeting, and that is a different Zoom meeting. So you can wrap up this session with your closing remarks.
Okay, awesome. So thank you everyone for coming, and be sure to let us know if you have any comments or questions on how this went. Hopefully next time we do this, it will be in person. yeah, so just let us know if you have any questions or comments on it. Thank you so much for coming. I know at least Jessica shared her information in the chat if anybody wants to reach out to her with for any further questions. So unless there's anything else for me, I'm going to go ahead and close out the session and wish you guys a great weekend.
Thank you.
Thank you.